Hello, everybody, and welcome back. I'm Srushti Chipre, and this is the Purple Science Podcast. In today's episode, I'm going to introduce you to one of my dear friends. Rachna is a master's student in Biological and Biomedical Sciences at University College Dublin in Ireland. Those who know her know that she's always beaming with happiness, especially when somebody talks about science. When we first learned about CRISPR, we were intrigued, we were curious, but her eyes were sparkling with excitement. For her, it was the moment she knew exactly what she wanted to do with her life. A little context here: CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. This is basically the defense mechanism that bacteria uses to fight off viruses, just like we have our immune system. What it does is it cuts off the viral DNA and prevents the virus from replicating further. Now, scientists have also figured out how to use CRISPR to edit the DNA of other organisms, including humans. Naturally, this has opened up a whole new world of possibilities for medical research and gene therapy. This technology can be truly revolutionary. It could be used to treat diseases like cancer and sickle cell anemia, and it could also be used to create crops that are more resistant to drought or to pests. Possibilities are really endless. Of course, like any other technology, it has some downsides and some ethical concerns. For example, the possibility of making designer babies, that is, babies with certain characteristics like a certain hair color, a certain eye color, and so on, without their consent. However, that is for another episode. Let me know if this intrigues you, and I will get into the details of this. I'm linking down a YouTube video below that gives an amazing gist of what CRISPR is and what it can do. For now, let's listen to Rachna and her amazing adventures in science.
Yes, so Rachna, right from the beginning, you were somebody who has always been excited for research and science in our group. What got you interested in research, first of all, and then what drives you today to continue it?
The one thing which keeps me motivated that whenever I was in the lab, I was the most happiest person. And I think that thing started since I was in my high school. We were doing some silly experiments in the lab. And one day I was in the lab and we were just looking curd under the microscope, and our teacher, like my biology teacher, she told us, just look how the microbes, or maybe to specify the bacteria, how it's moving, the live bacteria. And then I was just looking that under the microscope, and I did not go back to the class. I was sitting there for almost like 4 hours, which is 4 periods in school. And then one day, like, everybody asks you, what do you want to become? And I think that one experiment made me realize that I love to be in lab. No matter what I will be doing, but the one thing that keeps me happy is to do experiments and just observe things which you can't even see without a microscope. And I never liked botany. Imagine, today I'm working with plants, but I never liked botany before. Maybe something must have happened to that 17 or 18-year-old girl back then. She hated plants, not to, grow them in her garden, but to work with them, to understand how they grow, to understand how they function, and everything scientifically. I don't know what happened, but then zoology became my favorite, to just work with animals and how they grow, how they function, how can I use them. And then slowly my interest from NEET or MBBS thing deviated to research in scientist. So there's this one typical thing that usually happens in South is like either become a doctor or an engineer. And I was not that person to, just run behind the crowd. It was like, I will go in a different way, so let me just do research. Well, I didn't know the real meaning of research back then, but today I know, and I am happy for that.
That's really nice, Rachna. I think we know that your love for science originated in school, which is a great thing, I feel, because most of the kids don't know what they want to do, and the just the curiosity and the sort of, maybe not clarity, but the curiosity of this is something I
Yeah.
want to know about is definitely amazing. Would you also like to share some experiences or incidents in your undergrad days that sort of shaped your or are shaping your life in science today?
Wow, such a beautiful question. Well, if I did not have the experiences what I had in my undergrad, I would say that I did my undergrad in BSc Genetics, Microbiology, and Chemistry from St. Anne's Degree College for Women, affiliated to Osmania. I would say that, but no, I'd say I did my undergrad in Samyojana. And as I can see on your screen, it's like, Samyojana transforming undergraduate biology education. Yes, that's what I would say. That I did my undergrad in Samyojana and that changed completely. So let me just get into my first year experiences. First year was filled with curiosity and not clarity, like you just rightly said. But we had this 10-mark so-called assignment from Department of Microbiology, and that assignment turned out to be, like the real turning point in my research career. And the program called ICB phase 1, it is like integrating concepts of biology phase 1, where we were asked to read a research paper, and a first year student looking at a scientific research paper which made no sense. It was full of Greek and Latin, where they were talking about pancreatic ductal adenocarcinoma, PDAC. Our fuses were completely gone just by looking at it. And we struggled to understand. Well, I struggled a lot. And then I learned the basics of how to read the paper. And it was a challenging experience though. But it fueled my passion for science and research. And then I got to know, okay, the scientists do research and they don't find any other nice terms or easy language. So that is why they write complicated stuff in terms of a research paper. That was my understanding at that level when I was a first year undergrad. And then later on, we did read the paper multiple times to understand what's going on in that. We tried to understand the key concepts of it. And then there was a task called how to make a toast. So there were again different depictions of, the procedure of how to make a toast. We did that in our class thanks to our mentor who was conducting that workshop or program. And after that, we made our first poster, which was a cartoon poster. So it was just like, the unserious poster where a team is, trying to understand what we are making and what we are understanding. And we just dumped everything in that cartoon poster. And later on, our mentor was like, okay, you're not supposed to present this. He was like, it just looked childish. And he just told one statement that, would you like to be called as cute undergrads or the real professional undergrads? And then somehow that triggered us, and we looked at one of the scientific poster examples and we made exactly the same. In fact, I should say we copied the same poster but with our content. The design, the structure, and everything we copied
Mhm.
in a team. And I think this experience, it just sparked our interest in reading research papers and understanding their importance. And after several drafts of the same poster, I would say, the final poster that we presented at Dr. Reddy's Institute of Life Sciences, we were nervous. All faces were question mark. I was like, oh my God, who will ask me a question? What will I answer to them? I don't know. I only know what is there on my poster, but nothing else. Everybody were, the top notch researchers at drills. And well, it was a nice experience and we received valuable feedback from them, and that made our science better, I would say. And then again, back to the same college routine. We had our undergrad and everything, and then slowly we were like, this is not gonna work, we need to do something better now. Like, we were reading research papers, we were not getting the same kind of experience at college. So again, there was this another phase that was coming in our undergrad life, was phase two, which is called as integrating concepts of biology phase two, but then with a little more hands-on experience to research, where we were exposed to do, I mean, it was like a 14-day workshop in college. And by the time first year was almost to an end. And at that time, I think it was in January 2020, where people were talking about coronavirus and stuff, but we were all talking about 16 SRRNA sequencing workshop. And during that workshop, we were just staying back after college, and we were like, we're not going home because we want to do this workshop. So we did not care that we had to go home and things like that, while other students were just leaving home because, their buses were getting late and stuff. But we were like, no, we are just staying here and we are doing this. And we were completely immersed with the workshop activities. And then I realized that how much thought goes into that experiment. To design one experiment, you need to think a lot. And I was like, okay, this is something that I should be very careful when I become a scientist. And if I say this loud, that when I become a scientist and I should look into the very details of each experiment, it is wrong to put in that way. It's like we are already scientists and we are thinking in that way to become a better scientist. And then my curiosity led to ask questions during the workshop, and whatever is required to excel in that field, it made a difference. And with that real understanding in the workshop, I presented another poster at drills. And well, it was a solo fight to present the poster at drills because of lockdown and stuff. My teammates couldn't join it, so I had to do it by myself. And it's nice to know when you are discussing in teams, their point of view. So it was great to inculcate their views as well when I was talking about the poster. And suddenly 2020 March, 21st March, it was lockdown. As soon as we presented poster, we went back home and we stayed at home for 2 years. And then I think the real turn that took in my life, I would say. Until then, it was all the things about science, research. These words were all vague, even until then. The only thing in mind was I wanted to do research. I don't know where, what, how. I wanted to be in science for my life. I don't know how, where, what. The same questions. But again, there was lockdown. You got holidays. You need to do fun and enjoy life. Just relax on your bed, eat Maggie, and then, just sit and talk to your friends for hours. Nobody is there to disturb you. I mean, the biggest and nice part was to not to go to college. We were at home 24/7. We had our own laptop, tablets, lucky enough, privileged enough to have those things at home and use them. Watch movies. Well, Netflix and Amazon Prime is there. Watch it, use it. I did that for like 2 months without, I think I should say I wasted full time for 2 months to watch all of that, but it was needed somehow. And then I was like, okay, I need to get back to my work. That means I need to get back to science. And apart from science, there's this social life also, which asks you to do all these things. But then science is like, okay, you need to come back to me now. And that's how I'm waiting for you. So, and then I went back to science, and then I was like, what should I do now? It's lockdown, no college, no work. What can be done in lockdown? And then there is this wonderful program that pops into your life so smoothly, and it became your life for 1.5 or 2 years, I would. And that is why I told I did my undergrad in this program called Samyojana. And it was a great turning point for me. And that moment in my undergraduate journey was when this program gave me that, much needed clarity and exposure to the world of research. So, through the process of reading presenting research papers. I think, but personally, every 2 weeks, we had to present a paper in this program. And I worked in team, and we were receiving critical feedback from other teams. And I gained a deeper understanding of all the research methodology that I was reading. It was all new, Greek and Latin. I haven't read that before. I did not even know that I'll do that work, but then somehow it was making sense. And despite being a part of that program, I did not limit myself from just the academic works too. That program was there. I mean, I meant to do it anyway. But apart from that, I was also involved in multiple other things, and that is the biggest part of it, that it did not stop me from joining or, doing or involving myself in different activities, like starting my page on Instagram called Science and Abstracon, or talking to different researchers or PhDs from different part of the world, making new connections on LinkedIn, connecting with people just like that, talking to them about science, what they think about science, so that I will get clarity about my own science. And so all those things were going on along with this program during that time.
With the amount of people around us telling
Yeah.
how they are so burdened by their academic pursuits and challenged by their degrees, your
Mm.
joy of learning that you experienced, especially during the lockdown, is already a breath of fresh air, I think.
Well, I'm privileged to be in that program, I think.
Yeah, but since you mentioned about this, which I think I'd love to ask, with these experiences that you had with the
Mhm.
program you talked about, and then the workshop you attended, or whatever lab experience
Yeah.
that you had, now that you're doing your masters, what are the obvious advantages that you see yourself having as compared to the other people who might be struggling with some things? So, are there any things like that, if at all? And if there are,
Yeah.
can you shed some light on that?
So there's this one very important thing that I would like to highlight is I told you that I was reading research papers in that program for like almost 2 years, like if you take. And the amount of time that was invested in reading research papers as an undergrad was worth it, actually worth it. Without that experience, I would not have been in peace today doing masters. Because every day in masters, you have to read so many papers, do your assignments without understanding the level of the research that the scientists are doing and the efforts they are putting in writing those. And you will also figure out if that content is right or how and what not in that research paper. You will have so many things to look into it. So the amount of time that I already spent in reading those research papers, now it's just piece of cake for me. And a cherry on top to just go through the figures, summarize it, and you're done reading that. But when I see other students struggling with the same thing is concerning me a lot. I think they might have not got the same experience as me, but they were not even introduced to, reading papers or doing such stuff. And that is one challenging thing for them, because I did help few of my peers with reading papers, understanding them, and that was a nice group activity. But always being in groups at master's level is not, I mean, you the time that as an undergrad you can spend, you will not have that time as a master's or further as you go into your higher studies, I would say. And well, currently I'm doing my master's in Dublin, and if I did not have those experiences as an undergrad through the program called Samyojana and experiencing multiple aspects of science, I don't think I would have been so happy today. And well, I would say that apart from Samyojana, if I had other things also, which college would have given me, would have been cherry on the top. But okay, that did not happen. And now I am learning as a teaching assistant here in my college, and it's nice to ask questions all the time, and that's what they expect here. And asking a question is not like you are an idiot. In fact, if you don't ask a question, you look like an idiot. At least people who are asking questions are trying to understand stuff, and who are not asking questions, it's like they don't even care.
So you brought two very important points here. One was that asking questions and being curious and conversations, more than anything else, should be a part of science. These are how science is done. So that's very true. And when you said that the most valuable thing that undergrads have and that we underestimate is time,
Yep.
because as we go further ahead in our journeys, we might not have the kind of time for, going over as we used to do, like sentence by sentence and concept by
Mm.
concept, and having those discussions. That wouldn't be possible for them. And that is very important when you start your journey in the science. So that is very true. Coming to your because you mentioned that you always wanted to pursue your higher studies abroad. I'll start with, what are the kind of challenges you're facing as a young researcher studying in a foreign country? And then what of these experiences have helped you in this journey now?
Okay, so challenges coming from India, a pampered kid, the first thing. Never did the household work, the second thing. Always parents have taken care of everything. I've never taken a responsibility apart from science that I've done in India. Here, I would say that I'm not struggling for anything, to be really honest. It's just that my excitement that's keeping me motivated always, and I don't want to lose that. And all of the experiences that I've got as an undergrad, I think that is somehow fueling my interests, and it's paving a way for me to move ahead in taking decisions. Today I am with much more clarity in my head, and I can make decisions. Best part is I know what I want to do in life now. I know if I am good in the lab or in the industry, and I know myself better, and that is why I am not struggling.
Yeah, so can you also tell us about your teaching experiences and how you keep that balance between your research work and your teaching work?
Okay, so teaching just came to me as an opportunity. So I am teaching at my university as well as in one of the institutes in Dublin. So at my university, I do lab demonstration for undergrad students, and through that, I learn a lot. Because most of the experiments that undergrads do are repeated at master's level as well. Because the equipment, how to handle those equipments, how to design an experiment. It's just brushing up my basics, I would say, when I'm demonstrating them. And when I'm teaching at another institute, as you know, the biology teacher, so called biology teacher, there, when I taught them about plants, I told you, right, in the very beginning that I did not like botany at all. And now I am working with plants completely. And to know how plants function, what are the different parts of a plant scientifically, and what each part plays a role in photosynthesis, for example, or the development of the leaves, or how they react to different environmental factors and stuff. When I taught these things to my students, I would not say like taught, but when I discussed all these things with my students, I learned. And I was like, wow, I'm understanding my own research. So it's like I am discussing, I am balancing my level of understanding when I'm doing my research. So my basic for my research are established while I'm teaching. And when I'm going a bit deeper, I am not losing my basic understanding. And that is how I am easily able to research in my field. And currently I'm doing research with cannabis, and my aim is to create a carbon neutral society by using the very fascinative tool and very interesting, which did not allow me to even sleep when I got to know about is CRISPR, and it is a gene editing technology. And using that, I will be making knockouts in the genes of cannabis, and I will try to modify the plant architecture that will serve as a model for environmental studies. And in addition to that, I'm actively involved in science communication, and I love to interact with people.
Great work, Rachna. I think you're doing some really interesting work in your master's project. When you said that CRISPR has given you some sleepless nights full of curiosity, we know that that is very true. I think we were introduced with CRISPR
Mhm.
almost at the same times, but you were one of the very few persons who was so obsessed with it and who stuck with it, who has been working on it even now. So tell us about your first introduction to CRISPR and then the subsequent journey of falling in love with it.
Oh, wow. I love this question. I was waiting for this to start. And okay, well, CRISPR, it was introduced to us in 2020. Okay, and we were told that there's this interesting technology and where you can edit anything you feel like to in the lab, and you will see those results soon. And I was like, okay, there's something very interesting. And then suddenly we read one paper in a team, and in Samyojana, okay, going back to Samyojana, so I was a part of a team called Clelishers. So me and my friends, we were reading this article on base editing. Along with that, in the last para, they were summarizing about CRISPR. Okay. What happens was, we didn't know that bacteria had its own immunity called CRISPR, and we always thought that bacteria is so poor that viruses always kill bacteria. And one day we read about CRISPR and we say that bacteria is able to fight against viruses. That something got stuck in our head, and which was not introduced to us when we were studying about the lytic and lysogeny cycles in our undergrad days in the classroom. They only used to say that, okay, bacteriophage infects the bacteria and so on, the cycle continues. But we were like, at least you could have given us some, maybe not in detail about the CRISPR, but the different ways how bacteria can fight against viruses. And that's how the discovery happened in 2020. And we were reading papers related to CRISPR in 2021. And that one thing, I was like, okay, there is something. CRISPR is very interesting. The long full form called clustered regularly interspaced short palindromic repeats. And then I think I decided that I want to work with this. And that one thing is like, I want to work is like, I really want to be in that field. And then slowly I started reading about CRISPR, the papers that were published, and wherever I used to see about CRISPR, I'm like, I'm reading that. Please, you others are not supposed to read because I'm meant to read that. So I was thinking in that way, and most of the times, whenever I read about CRISPR, I feel that there are so many things. It has endless opportunities to work in that field, and slowly that area is emerging. And
What are the applications of CRISPR that excite you the most?
okay, so the one important application is gene editing. And I think CRISPR technology has made it easier, faster, and even affordable to edit the genome of any organism, I would say. And the one thing, when I heard the talk from Jennifer Doudna, like one of the TED Talks, she talks about editing humans and making designer babies. And I was like, wow, I will do that one day for sure. She's just talking. I will do that in the lab one day. And I think that one thing is very interesting. Apart from that, there are so many things. Like, I'm working with plants today. And I think in CRISPR can be used to, create crops that are more resistant to pests and diseases and more tolerant to different environmental conditions, like drought and even the extreme temperature conditions. And they can be more nutritious when I use CRISPR in the plants. And this could, address so many food challenge, like, the food security challenges in different parts of the world. And Gene editing is one such application which is, aligned with agriculture, as well as even in animal models. There are different animal models where CRISPR can be used, and it will help to create animal models of human diseases, where human diseases can be studied in those animals, like zebrafish is the best example. Mice is like always there, and I think these can be used to study the mechanisms, and it can be further tested for the potential treatments, as well as this has, and then later on the drugs can be discovered and stuff with this.
This is really super fun. It's always great to listen about CRISPR from you because your excitement makes it all the more fun. With the already fun undergrad experience that you've had, let's say you had full-time lab access and enough
Wow.
resources, whatever resources you need in
Mm-hmm.
your undergrad,
Mm-hmm.
what CRISPR experiments would you have liked to design and perform in your college lab?
College lab. Okay, well, going back to college. College was one thing where you were not supposed to touch anything, okay? You were only supposed to take those things which are already kept by the lab attenders and nothing else. But I think by the time I was in my final year, somehow we were getting into the labs, we were doing stuff and all. But luckily, I mean, I wouldn't say luck, but yeah, okay. Luckily, I was a part of this internship at Dr. Reddy's Institute of Life Sciences, where we were trying to, make transparent zebrafish lines. To that,
Can
we
you
wanted to,
quickly tell us what are transparent zebrafish lines, and then what does it mean for us to have these particular, like, why would we want zebrafish to be transparent?
Okay, well, so zebrafish is the animal model, a type of animal model where it does help in studying rare diseases. So for that, our objective was to just using CRISPR to create a knockout one of the genes called as MIT-FA, which codes for a protein called melanin in zebrafish. So basically, that is a transcription factor in zebrafish. MIT-FA is a transcription factor, and when we use CRISPR, it will knock that out of zebrafish. So when we are knocking that, it creates the transparent lines, and that is how we could visually see what is exactly happening in zebrafish.
Why do we need to see that?
It makes it easier to study as it is growing. If you want to study about different cancers, then you can clearly see from where the cancer is growing in the cells, right? So that is one reason I would say.
Oh,
And in that experiment, going back to the CRISPR basic, we attempted to design guide RNAs, which was a bioinformatic experience where we were introduced to a new website altogether called Benchling.
mhm.
Through it was all fancy, okay, and but then we learned how to design primers and then guide RNAs. Not very perfect in that, but today I am able to do it in a much better way. But without that experience, it would not have been easy to do it.
I think first you've heard a lot about CRISPR. You might have seen images, you might have gone
Yeah.
through a lot of resources. What did you imagine CRISPR was versus the first time when you stood in lab and you performed this experiment, when you had to use this tool? What were the revelations or realizations that you had? And just tell us what happened when you first entered the lab and you did this first experiment.
Okay, so CRISPR, as such, I was reading a lot about it, and through images, it was clear that it is a sequence. And I was expecting to see the same colorful things happening in the lab. But no, everything was transparent in those small test tubes. I was like, okay, it is transparent. You don't know what's happening. It's all unknown. It's a mystery. You're just labeling them because you know that you have put something in that, and just do the experiment and see the result. It could be DNA sequences, it could be CRISPR guide sequences, the guide RNA sequences. But when I see them in the lab, everything is just transparent, not even white color, transparent. What do you think a student should understand just by looking at that? Nothing. But then slowly, I think I was used to it because I was seeing them almost every day. And then I realized that with the level of imagination you understand concepts, with the level of understanding that you go deeper in that field, with that imagination and with that level of understanding, you should be able to analyze the results which are transparent enough in the lab. And I think that is the one way to, just look at those DNA sequences or the CRISPR that is introduced into the genome, and wherever it makes the knockout, to, just read those gels. It made sense after a much longer time. For me, it really took some time to understand the bands on the gel, because I used to think, oh my god, this is just one black color line on the gel, on the piece of a gel. What should I do? What should I infer with this? And I was like, okay, let me go back to my basics. And somehow I would say, by constantly looking them for almost every day, reading the same thing every day, I understood. And the next time I imagined how a CRISPR look, I was only imagining the CRISPR sequences that I see on paper. The sequence, at the sequence level, what should happen, the DNA sequences, ATGCs, I was imagining that in my head. I was imagining how they are bound to each other and how they work. And when you're introducing CRISPR, how it breaks. Because there's so many visual effects on YouTube. Those are made for your understanding, but it happens in all those transparent liquids in the lab. And it's hard for the student to figure out what's happening. You see something and you visualize something. So it makes a big difference. And I think that is where one should be really clear about what they are looking at and what they are inferring at. So it will make a difference.
It's
Clarity.
really beautiful how you said it, Rachna. You said that, the first time you saw it, you saw all colorful pictures and processes happening, and then in the lab it was all transparent. But then the more often times you've like seen it and you keep seeing it, keep reading about it, you again start to see those colors and all those bands start making sense to you, which is I think beautiful.
Yeah, and I think, even science is like that. In the first instance, you will not understand anything, but you love it
Mhm.
because there's something mystery that's going on. But when you talk to it, when you read about it every day, you will just love it.
That's
And and I would say the field is like that. Who would not love biology?
Well, who
I
are
love
some
it.
people you
Biology.
look up to in your research career?
Okay, there's so many people, but the first person I would say is he was my mentor. He's still my mentor and he will be my mentor. He's Dr. Anil Kumar Challa, and I think the way he portrays science was next level for me as an undergrad. And the way he guided me to look in that field, or to pursue career in that field, was very, very, very fascinating. And the second person would be my mentor from UCD, because the way I look up to them, I'm like, I want to be like them. I mean, the way they teach, the way they talk to you, the way they interact with students is different. And I'm like, okay, you should look up to them. And well, of course, the great people who have discovered DNA, James Watson. Well, I don't want to be like him, how he was in his social life, but then the curiosity that he had, I want that. I always want that. And of course, Jennifer Doudna. I should take her name. And Elizabeth Bick is one more person. Okay, well, I only open Twitter for her and nobody else. I only go to Berkeley Doudna's lab only for Jennifer Doudna to see what research she's doing, what are the recent updates. And I only have their notifications on my Twitter and nobody else's. And the current research that I'm doing is on cannabis, so I only have those notifications on. And there are very few researchers who are in that field, so I'm excited to use CRISPR there again. And of course, I'm looking up to Richard Feynman now. He is completely a different character altogether. But I think that's how the fields in science are. And it is a blend of different mentors and different scientists' stories and everything that is keeping me alive and keeping me motivated to do that work.
All right. One last question that I ask everybody.
Yep.
With the experience that you now have, and I
Mm-hmm.
think you have a lot of fun experiences, let's say if you were to relive your undergrad days, what would you do differently?
Okay, I would not go to college. I would be exploring to different institutions. See, it
I
was
was I was
so.
expecting a very Rachna answer, and you did not let me down.
Thanks for that. See, I did not even have to think about that. It was so spontaneous.
Mhm.
And definitely, I would, me and along with my friends, even if they say they want to go to college, I would say, no, we are not going to college anymore. We are just exploring stuff. We are talking to people. We are going from lab to lab and no college.
That was it for today's episode. Thanks for listening. As I always say, we thrive on your feedback. So keep sending in suggestions about what you want to listen to and who you want to listen from. I'll be back with another story next Tuesday only on the Purple Science Podcast. Thanks for tuning in to the Purple Science Podcast. We share stories every Tuesday. Our inbox is always open for your feedback, and our DMs across all social media platforms linked in the description are open to your unique stories. If you know somebody who might benefit from this, do share the podcast with them and spread the joy of learning.